Thank you very much, Kevin. And I'd also like to thank ACAMP for inviting us to present our work here today. Good morning, ladies and gentlemen. And I'd like to get started with introducing the term lab on a chip. I know Kevin did a great job introducing microfluidics and lab on a chip, so my work here is relatively simpler. And I will move on to talk about the surface microfluidic technology that we offer or we want to offer. And the microfluidic devices that we fabricate and produce are based on the concept of dielectrophoresis. So a little detail about what dielectrophoresis means. I will then move on to the applications that we have today, that we can demonstrate today, and also the potential applications that we offer in the field of cleantech and also in the field of biodiagnostics. I'll close my presentation with a little bit of the future work and with acknowledgements and some of our achievements. A lab-on-a-chip device integrates several laboratory functionalities on a small platform. These functionalities can range from sample aliquoting, sample dispensing, to transport, manipulation, mixing, achieving combinatorial chemistry, detection and diagnostics, everything on a small platform. Some of the existing lab-on-a-chip devices I've just shown on the slide. So, one can divide lab-on-a-chip devices in two clear streams. One is a closed-channel lab-on-a-chip device, and alternatively, what we call surface microfluidic device. A closed-channel lab-on-a-chip device incorporates, these are continuous flow-based devices. They use microchannels, high-pressure valves, pump and tubing arrangement to create flows of samples and reagents in very low volumes, in microliters to nanoliter volumes. And they conduct combinatorial assays, detection and diagnostics on small microfluidic platforms. Alternatively, what we are proposing or what we propose is a DP-based surface microfluidic technology. These are precision droplet-based devices, and they can dispense sample and reagent droplets in the range of nanoliter to sub-picoliter volume with quite a good precision and accuracy. And they can manipulate these droplets, they can transport them, mix them on these microfluidic platforms. Now, the reason why we choose these surface microfluidic devices over these closed channel devices is because these are precision droplet-based technology. So we further reduce the requirement of sample and reagent volumes. So the dead volumes are low, and since there is less exposed surface area, so we try to minimize the adsorption and contamination issues, which is essential if you're doing detection. And also because they bear an open surface, it's easy to do integration of hybrid integration of actuation or sensing units on the microfluidic platform. So how does surface microfluidic relates as a clean tech? Well, it is cost effective, as Kevin explained, because these are miniaturized devices and they can be bench fabricated, bench produced, so the production costs can be lowered when they are produced in bulk. And since they offer or promise a capability of hybrid integration to devise a very portable and efficient device, something like a handheld device that can do or replace most of the laboratory systems or the pharmacies that exist today in future, of course. So they definitely, by miniaturization, they will reduce the cost and they will be efficient and effective. And they reduce the requirements of samples, biological or biochemical samples. And they hold promise to replace expensive off-chip components. Okay, dielectrophoresis is an electrokinetic phenomena which we use to manipulate dielectric fluids or dielectric particles by applying a spatially non-uniform electric field. I won't go in much of the physical principle or the details of the actuation methodology. What I will rather do is show its application. The application of dielectrophoresis in surface microfluidic can range. The first one that I've shown here is its ability to dispense variable volume and controllably position droplets in the range of nanoliters to sub-picoliter. The scheme is we form a liquid jet and we control the breakup of this liquid jet to form arrays of droplets. The actuation process is a high-speed actuation process, and as you can see some of the details here, the droplet volumes can go very low in few picoliters, And the actuation rates are very high. It only takes like few milliseconds to dispense arrays of droplets with a very good flexible control. The second actuation scheme is a droplet manipulation scheme. So what we can further do is controllably move these droplets to different on-chip locations. And this comes in picture when we try to develop on-chip mixing systems or on-chip detection systems, like the one shown in here, like a multiplexed reaction chip, such as this one here. It can handle four different samples or reagents, and it can do combinatorial chemistry. We have demonstrated several biodiagnostic systems based on. Sorry, the video didn't come up. Okay, anyway, moving forward. We have demonstrated biodiagnostic systems that can quantify biosamples in the range of few femtograms. That is a very good precision of detection that we can offer, and it's comparable to the microchannel. It's better than the microchannel technology and comparable to the off-chip technology that is available. The macro detection schemes. The other big application that we offer is a precision emulsion dispensing technology. We have a device, we have an actuation methodology which could be used to form numerous arrays of very precisely positioned emulsion droplets. These could be monolayered emulsion droplets or single emulsion droplets, or they could be double or triple higher multiple layer emulsion droplets. Now, normally, to dispense emulsion droplets, first nobody can achieve a precision dispensing of emulsion droplets. They have no control on the population of the dispensed emulsion droplet arrays, and the existing technologies are complicated and expensive, like the high energy homogenizers or the sonicators that are available today. What we offer is a simple microfluidic chip that can dispense arrays of precision droplets, emulsion droplets, single or higher order emulsion droplets, which have several applications ranging from healthcare. They could be used to form vesicles. We have already demonstrated its application to form lipid membranes and biosensors. They could also be used in cleantech because they hold the promise to form laser fusion targets in renewable or alternate energy systems. Other potential applications that we can offer through our surface microfluidic devices in cleantech is a sub-nanoliter droplet dispenser. The existing dispensing schemes that we have, the pipettes that we have, can only go to 0.5 microliter, and that is it. What we can offer is a sub-nanoliter dispenser that can go as low as few picoliters, and it can digitally control the dispensed droplet volume. So imagine a handheld device with which you can dispense few nanoliters or few picoliters of samples and reagents with a quite good precision. The second application, of course, is because our device offers a functionality of dispensing arrays of droplets with a good precise dispensing. So the microarray droplet dispensing scheme can replace expensive robotic arms, which are used for microtiter plate assays or for doing microarray technology assays. The third application is to use these reaction chips like the one 2x2. The video didn't play, I'm sorry about that. These reaction chips that can do assays on a platform, multiple assays on a platform. And by doing that, they can replace laboratory-based testing facilities like blood testing, blood glucose level detection. And another application is when we promise that we can dispense ultra-fine precision emulsion droplets, they can replace the homogenizers or the sonicators, the expensive devices, and at least they can replace them for microfluidic applications. The cleantech applications that we have brainstormed on we haven't actually demonstrated them, but we hold the applications that we feel there is a potential application in these fields. The first one is to detect heavy metals in water sample, to detect very low amounts of carcinogens in water sample, to do trace element detection, and to detect biological pollutants. And the good thing is we are very good with low detection thresholds. So we can detect these trace elements as low as 10 to 100 parts per million, and microorganisms like a viral DNA or a bacterial DNA as low as few femtograms, that is like few hundred molecules. So it's a very precise detection scheme. And yeah, we can also study shelf life of food samples because by providing these emulsions, we can create a controlled environment that could be used to study life cycle of various food grades. And the target, my research was focused on showing the capabilities of the microfluidic platform. But as a PhD project, and our future target is definitely to integrate the entire system and to create a device which will have a hybrid integrated sensing unit. Our colleagues have demonstrated an integrated CMOS optical sensor that can detect and quantify the amounts of microparticles in a specific fluidic sample. We also want to integrate the actuation unit on chip if possible and create a handheld package device that can be used for applications ranging from microfluidics to cleantech. I'd like to acknowledge the support that we have got from our industrial collaborators and granting agencies, including UTI. And most of the fabrication work and the design work was done by our group, by ourselves, but was helped and supported by NanoFab at the University of Alberta. And I'd like to thank Dr. Kaler, my supervisor, for providing me this opportunity to present our work in front of this audience, and also my past and current group members. Thank you.